Hi, this is Dr. Yunong Liu. In this short video, I'm going to very briefly go through some of the quality control that we normally look at after receiving the single cell or 10x derived single cell data. After we receive the FASTQ files, and the first thing that we will do is to use Cell Ranger, this particular software provided by 10x to do the sequence alignment and the gene expression calling. So we will, after that, we will generate a file like this. And let me go through a few key points we want to look at in this particular file. So the first one is the estimated number of cells. And as you can see, in this case, it's about 1,800. And as you know that we are aiming at, sometimes in most of the experiment, we'll aim at 10,000. But this will let you to decide how many cells that we really identified in this particular experiment. And the next one is the number of arrays per cell. So you can see a lot. And then the number of genes that are being identified per cell. And this is one of our earlier data sets during the training. And then we will also look at the number of total arrays. In this case, is about 287 million arrays. And then you can see that 97% are having valid barcode. Remember that barcode is the one that corresponding to individual cells. So for the sequence arrays that share the same barcode, they are considered coming from the same bubble in the previous lecture, but essentially the same cell. And over 99.9% that is having the valid UMI. Or unique molecular index. And another information I often look at is this sequence saturation rate. And this is a number that somewhere you can see, of course, it ranges from 0 to 100%. But the larger this number is, that means your system is being saturated, which means if you sequence more to the same library, you wouldn't be able to identify more genes for the cell. Normally, we want to control this level at certain level, about 30%-ish, and that would be a good experiment. Otherwise, that you don't want this number to be too low, that you know that if you sequence more and you're going to get more information, or you don't want it to be too high, that indicates that you waste a lot of sequencing rates already. And all the remaining numbers, it gives you different level of quality control. So you can see it's all over 90%. And after sequence alignment, that we also want to know that what are the percentage of the rates that being mapped to the genome that for the species that you are sequencing. In this case, 95% are mapped to the human cells, a human genome. That is really good news. That's indication that you got a lot of very useful data for your results. And the rate mapped confident to the genome, 92%, and to the intergenic region, 4.5%. If you look at this, and you'll have very good confident that you are sequencing the RNA molecule, because there's only a very small percentage goes to outside of the gene regions, which is the intergenic regions. You'll also see about 19% or 20% that goes to the intronic regions, and the remaining most goes to the exonic regions. And normally that we will see about 20% of rates goes to intronic regions. And however, for the nuclei sequencing, this number will go up a little bit, somewhere around 35 to 40% of rates goes to the intronic regions. Estimated number of cells, 1,800, and the fraction of rates in the cell, 96%. So we identified over 1,800 cells, and then you see the 96% of the rates actually are detected in the cells we identified. That is really very high quality data. That is something related to the figure above, and I will go back to that in a little bit later. And the mean rates per cell, and which is repetitive of this information, but you can see that all the numbers are being shown here are pretty good. So now let me go through this figure a little bit. This is something that I really look at when I see the data, the quality, trying to assess the data quality. In a short sentence, that I'm looking for this shape. This is called a knee plot. As you can see here, that in this part, oh, sorry, I did something. Let me refresh this. I forgot that I need to get the annotation going. Okay, so let me get to here. So this is the part that you see, this is a knee here. And what I want to see here is there's a gradual, it's pretty plateaued, it gradually goes down, but there's a sudden drop. So what this means is the X here is the number of barcode. So if you look at here, this is about somewhere around, let's see that if I'm moving my mouse over here, and then you can see about there's somewhere around maybe 5,000 barcode that we will receive the number of rays or UMIs that more than certain numbers. And if you look at this, and this tell me that about 100 cells that their UMI numbers we detected is about 100,000 rays, very excessive. And then this number will go gradually. So if we move to here, there is about a few thousand barcode that the number of rays goes beyond it. Maybe here is like a 20 or 30,000. So this is the number that is showing here. You want to see this sudden drop, because if for one scenario that I'm giving you, a very bad scenario, that your cells are all bursted, you basically, you just, all the RNAs are floating around within your tube. And then when you trap those bubbles, and each bubble will still, by random, will trap some of the floating RNA or ambient RNA in that. So in that case, that I will see something like this, and this will be gradually dropping and very much gradually dropping, something like this. If I look at this type of shape, that means that I'm not confident any of the bubbles trap the real cell, because it's not significantly higher than the random case, because you will expect to see this gradually drop anyways. But for this case, as you can see that there may be, here is up until here, those are the real cells. And after that you can see that the number of rays that being detected suddenly drop to a very, very low level, only a few dozen of the rays per bubble or per cell. And in that case, those are the non-usable cells. So in this case, that we will see that this sudden drop here, and then there is a gradually small ladder here, and then further drop off. So let me show you one other figure that I give you a very bad example, which is in this case. So you can see that I'm seeing here is like really the gradually drop, and I have not observed those sudden drop at a certain point. And this is an indication that the experiment is not giving me very good results. I don't know whether I explained this well. This is a complicated topic in this part, but if you have any questions, we can further discuss this particular Friday. I hope you enjoy it, and thank you for watching.